morning. Today our group will present about chromatography. First, let me introduce my group member. First, we have Nur Husna Izzati, Nur Rafika Najwa, Nur Fazila Tasha, and Putri Nur Aizira. Next, for content, we have introduction part, type of chromatography, theory, methodology, example of result, and last is conclusion. For introduction part, chromatography is a basic yet powerful laboratory technique and used to separate and identify components in mixture. In medical laboratories, it plays a vital role in analyzing complex biological samples such as blood, urine, and saliva. Through this technique, medical professionals can detect the presence of drugs, hormones, glucose, toxins, and other important substances in the human body. By separating these substances based on their chemical properties, chromatography helps doctors diagnose disease, monitor treatment progress, and ensure that medications are at safe, effective levels. Basic forms of chromatography, such as paper chromatography, and thin layer chromatography (TLC), are commonly used for quick and efficient testing. This technique is essential in toxicology, drug screening, and clinical research, making it a key tool in modern healthcare and diagnostic.
Let's begin by looking at basic type of chromatography: paper, thin layer, and ion exchange chromatography. Paper chromatography uses a special kind of paper to separate substances, mainly for analyzing color, such as ink or plant pigments. It's simple, cost-effective, and great for educational labs. Next, we have thin layer chromatography, or we call it as TLC. This method is quite similar to paper chromatography, but uses glass or plastic plates coated with a thin layer of adsorbent powder. It's commonly used in laboratories to check the purity of substances or to monitor chemical reactions. Next, we have ion exchange chromatography, separates by molecules based on their electrical charge. This is typically useful in purifying proteins or cleaning water. It works by attracting and holding ions with opposite charge to charge resin inside the column. Now, let's look at more advanced techniques such as high-performance liquid chromatography or HPLC and gas chromatography or GC. HPLCs use a high pressure to push a liquid sample through a column filled with solid material. The separation based on how the compounds interact with the material inside the column. HPLC is widely used in pharmaceuticals and food analysis because it provides high accuracy and precision. Gas chromatography, on the other hand, involve vaporizing and sample carrying it through the column using an inert gas like helium or nitrogen. It's perfect for analyzing volatile substances like alcohol, fuels, and fragrance. The compounds separate based on difference of their boiling points and how well they dissolve in the liquid coating inside the column. Lastly, we have column chromatography and affinity chromatography. Column chromatography uses a vertical tube filled with solid material like silica. A liquid solvent carries this mixture through the tube, and the components separate based on how strongly they stick to the solid. This method is used to separate large amounts of mixtures, while affinity chromatography is a very specific method. This separates molecules based on biological interaction. For example, a target protein may bind into a specific antibody or enzymes that attach to the solid phase. This method is ideal for purifying antibodies or DNA with high specificity.
Theory for chromatography is a technique first discovered by Russian scientist Twets. It used to separate mixture into their individual components. The process involves two phases, a mobile phase, like a gas or liquid that carries a mixture, and a stationary phase, usually a solid or porous material the mixture moves through. As the mixture flows through the stationary phase, the different substances interact with the material in different ways, mainly depending on their chemical properties and size. Because of these interactions, some substances move faster and some move slower. cousin, then the separation happen because the molecule are constantly moving between the two phase diffusing and interacting until they reach a source of balance. Each substance spends a different amount of time in each phase, which ultimately help to separate them.
I'll show you the methodology for a chromatography experiment, specifically using paper chromatography. This technique allows us to separate and identify the components of a mixture based on their different rates of movement through a medium. The objective of this experiment is to separate and analyze the components in a simple mixture, such as ink or plant pigments. The materials we'll need to use is chromatography paper or filter paper, a beaker or chromatography chamber, a pencil, a ruler, and a capillary tubes, a suitable solvent such as ethanol or water, and our sample mixer. Also, a support like a glass rod to hold the paper. First, we prepare the chromatography paper. We cut it into a size and draw a light pencil line about 2 cm from the bottom. This is known as the baseline, and it's where we apply the sample. We then mark the points along the baseline where each sample will be placed. Step 2. Applying the sample. Next, we use the capillary tube or micropipette to apply a small drop of sample mixture to each marked spot. It's important to let the spots dry and reapply if needed to get a concentrated dot. Step 3. Preparing the solvent system. We pour just enough solvent into the beaker so that the bottom of the chromatography paper will be submerged but not the spots themselves. Then we cover the beaker to help saturate the air with solvent paper. This helps improve the separation. Step 4. Developing the chromatograph. We carefully hang the paper in the beaker, ensuring that the spots stay above the solvent level. The solvent moves up the paper by capillary action, carrying the components of the sample with it. When the solvent front reaches near the top, we remove the paper and mark the solvent front with a pencil immediately. Last step, drying and analysis. Once the paper dries, we will see separated spots that represent different components in the mixture. To analyze the result, we measure the distance each spot travel and also the distance of the solvent front move. Using these measurements, we calculate the RF value for each component using the formula. These RF values can help identify substances based on known standards.
Now I'll briefly explain 4 types of chromatography used to separate mixtures. First is paper chromatography. It's used for colored substances like ink or food dyes. Spots appear at different heights on paper, and we calculate RF values to help identify each compound. The second one is thin layer chromatography or TLC. Uses a coated plate. Spots can be seen under UV light or with stains. It's commonly used to check sample purity and separate compounds by polarity. The third one is gas chromatography or GC, separates volatile compounds. Results appear as peaks on a chromatogram, with each peak showing one compound, commonly used for testing alcohol in blood. The last one is high performance liquid chromatography, HPLC, which is used in pharmaceuticals to check drug purity. It also gives a chromatogram showing how many components are in the sample and in what amounts. Each method is useful depending on the type of mixture and purpose of analysis.
Last, for conclusion, chromatography is a powerful method for separating and analyzing nature. It helps scientists and medical professionals identify different substances accurately. Whether it's testing food, medicine, or blood, chromatography plays an important role in research and everyday life.